Our last speaker of this session is Zeynep Gümüş, speaking about visualizing and identifying lung cancer risk genes.
Thank you. I'm going to talk about two separate research areas in my lab. One of them is in identifying risk genes in lung cancer, and the other one is in data visualization, and I hope to do both in 5 minutes. So we are interested in identifying risk genes for lung cancer with rare mutations. A lot of studies in GWAS has shown that there are common variants that predispose to cancer, but these usually have a low impact. However, rare variants usually tend to have stronger impact. So we're interested in such variants for lung cancer. Lung cancer is the highest leading cause of cancer, and non-small cell lung cancer is the most common subtype. If it's detected in early stages, lung cancer can be treated better, but if it's in stages 3 or 4, then the survival is not high. And if we can identify individuals who are at high risk of lung cancer, then we can identify them early on through screening, rigorous screening, or other chemoprevention methods towards precision prevention specific to their variants. We know that there are strong environmental factors, most importantly smoking in lung cancer risk, and therefore lung cancer is a little bit more difficult to study compared with prostate or breast, for example, where we have we don't have that strong environmental risk factors. So in our lab, we started a consortium with multiple investigators from several institutes within New York and Israel. And first, we decided to look at individuals with lung cancer in a enriched population of people with lung cancer in their families. And also, we then, based on these results, we also have a validation case control set to be able to identify individuals to validate the results that we have. So the objective is to find the equivalent of what we have in breast in the lung, and so far we don't have. And the best example for breast cancer is the BRCA mutations, and people, even through 23andMe, they can now identify if they have a BRCA mutation or not, and based on that, they can do something about it, which is precision prevention. Either get, usually, for example, the best example being Angelina Jolie getting double mastectomy, in order to prevent it before it happens. And if you look at Angelina Jolie's family history, there is a really strong signature through her family history, and she, because of her variant, her risk for breast cancer was really high. So we want to be able to get there with lung cancer, even though it's more complicated for the reasons I told you. Another example would be colon cancer, where people at high risk of colon cancer take a daily dose of aspirin, which decreases their risk. So if we know what is predisposing, then we can do something about it. So in our study, first we looked at squamous lung cancer cases, and for this we used the computational pipelines of Mount Sinai, looking at several thousand controls with hundreds of cases in our discovery and validation cohorts. And when we look at these individuals, we saw that individuals that we have in our cohort were mostly European ancestry, even though we had from Asian to African ancestry. And so when we look at that, as a group, we identify Fanconi anemia genes as predisposing individuals to lung cancer. So this is really exciting, even though it makes screening more complicated because these individuals cannot get CT scans because these are DNA repair-related genes. So we definitely need to develop new strategies to be able to help these individuals. And now we're doing a similar study with analyzing whole exome sequencing data of thousands of individuals, thanks to the resources of Mount Sinai again. and for lung cancer adenocarcinoma. And then in terms of data visualization, my group has developed a tool called Icahn, Interactome Came, for 3D visualization of networks. Here's one example. It's an example that we published in Science of a network, and you can look at it as a video. This is another example we have in Nature Neuroscience with late Pam Sklar, where you can again look at the interactions, and also we can also look at networks that have known 3D structure with our tool, for example, a network in the brain or in the neuron or any networks that you're interested in. As you know, at our Icahn Institute, we have high interest in networks. So we would love to collaborate with labs that would make use of our tools. So thank you, and I'm happy to answer questions. I have 20 more seconds even.